The immune system is highly specific for each invader, and that's because the cells of the adaptive immune response have unique receptors that can differentiate friendly bacteria from potentially deadly pathogens from their unique parts, called antigens. The key cells of the adaptive immune response are the lymphocytes, the B and T cells. B cells develop in the bone marrow, where they undergo a process called VDJ rearrangement to generate a massively diverse set of B cell receptors. The B cell receptor is essentially an antibody, except that it has a transmembrane part that goes through the membrane, attaching the receptor to the surface of the B cell. The B cell receptor has two heavy chains and two light chains. And the region or fragment of the B cell receptor that binds the antigen is called the fragment antigen binding or FAB region. The FAB region is where the ends of the heavy and light chains meet, and there are two FAB fragments on each B cell receptor. The remainder of the heavy chain makes up the constant region, or constant fragment region, also called FC. The two heavy chains are linked to each other by disulfide bonds, and each heavy chain is also linked to a light chain by a disulfide bond. Each B cell receptor has two identical heavy and light chains, resulting in two identical antigen binding sites. As the B cell develops into a plasma cell, the B cell receptor gets secreted as an antibody with the exact same antigen specificity. However, the heavy chain actually changes as the B cell develops. There are 5 major types of heavy chains, which encode the isotypes or classes of immunoglobulins: IgM, IgD, IgG, IgA, and IgE. These 5 are encoded by heavy chain genes which are referred to by the Greek letters mu, delta, gamma, alpha, and epsilon. When a B cell is first developing, it initially expresses the mu heavy chain, and as a result, all of the B cell receptors are IgMs that are on the cell surface. When the B cell finishes developing, it undergoes a process called alternative splicing. Alternative splicing is a process by which the cell expresses the heavy chain exons for both mu and delta, allowing for both IgM and IgD to be simultaneously expressed on the surface. At this point, the B cell is mature, but still naive. Having IgD on the B cell surface is like a young adult with a driver's license.
Smile, please.
They're able to go out and explore the world. In the B cell's case, that means all of the body's lymphatic system. But these cells haven't been exposed to much, and don't know how they'll react to foreign antigens. Once the B cell starts to explore the body's lymphatic system, they travel from lymph node to lymph node in search of antigens. Lymph nodes are scattered throughout your body, and each one is a highly organized structure where millions of B cells, T cells, antigens, and antigen-presenting cells pass through every day, kind of like a busy airport. Now, both B and T cells, as well as antigens and antigen-presenting cells, all need to enter the lymph node. There are 2 ways to do this, depending on if the cells are entering from the lymphatics or the blood. If the cell is already in the lymph, then it can enter the lymph node through the afferent lymphatic vessel. If the cell is in the blood, then it can use the high endothelial venules, or HEVs. HEVs are special endothelial cells found at post-capillary sites of the lymphoid organs. These endothelial cells are more plump than the average flat endothelial cell, and they also express adhesion molecules like L-selectin. When lymphocytes pass through the HEVs, they can bind to and wiggle through the endothelial cells and slip right into the lymph node. When B cells and T cells get into the lymph nodes, they first enter the paracortical region. The T cells stay there, while the B cells migrate to the neighboring cortical region of the lymph node, where they form the primary lymphoid follicles. If a B cell gets activated, it starts replicating within a follicle, and it forms a germinal center. A follicle with a germinal center is called a secondary lymphoid follicle. Various antigens enter the lymph node through the afferent lymphatic vessel, and they percolate through the paracortex and through to the follicle. It looks kind of like a game of Plinko, where the antigens get to interact with a lot of B cells in the follicle. B cells, unlike T cells, can recognize a wide variety of antigens, including peptides, carbohydrates, and lipids in their native form, meaning that they don't require antigen-presenting cells to process or present the antigen. In order for the B cell to be activated, the antigen has to cross-link the B cell receptors. When two B cell receptors get cross-linked, their intracellular chains, the side chains Ig alpha and Ig beta, and CD19 all cluster together. Each of these chains have something called an immune receptor tyrosine-based activation motif, or ITAM for short. The ITAM is a conserved sequence of amino acids that includes two tyrosine amino acids. The tyrosines and the space between the two tyrosines is super important, because both tyrosines need to be phosphorylated for the cell to get activated. These two tyrosines within the ITAM region of the protein get phosphorylated by three protein tyrosine kinases, called BLK, FYN, and LYN. Think of them as a friendly little trio of phosphorylators. Once the two tyrosines get phosphorylated, there's a chain of events within the cell that ends with the activation of the major transcription factors, NF-kappa B and NF-AT. These transcription factors turn on the expression of genes that result in more cytokines, like interleukin 1, interleukin 2, interleukin 6, and tumor necrosis factor alpha. They also cause upregulation of anti-apoptotic cell surface markers, like BCL2, as well as the proliferation and differentiation of B cells. An entirely different, alternative way for a B cell to be stimulated is using the molecule CD21, which is also called CR2 because it was the second receptor identified for one of the complement fragments, C3d. In the complement pathway, a variety of complement fragments are produced, many of which can bind non-specifically to antigens. C3d can bind to an antigen and can then be bound by CD21 on a B cell. So if a B cell has a B cell receptor that's bound to an antigen, and it also has a CD21 that's bound to an antigen, then that will also initiate B cell activation. An activated B cell has one goal in mind: to differentiate into a plasma cell and secrete antibody. But there are 5 classes of antibodies that the plasma cell secretes. So initially, the B cell will differentiate into an IgM secreting plasma cell, because that's ready to go, since it's the basis for the B cell receptor. If, however, an activated CD4 positive T cell is present, it'll help the B cell to isotype or class switch, to change the isotype of the antibody it produces. Isotype switching requires a change in the DNA, and once the B cell switches its isotype, it can't go back to making IgM again. Now, B cells are one type of antigen-presenting cell, so they present antigen on an MHC class II molecule to a nearby CD4 positive T cell. If the T cell gets activated, it'll express the molecule CD40L on its cell surface, which binds to CD40 on the B cell surface. This causes the B cell to express cytokine receptors, and causes the T cell to secrete cytokines, instructing the B cell on the type of antibody to start producing. For example, if the T cell secretes interleukin 4 and interleukin 5, then the B cell will become an IgE secreting plasma cell. On the other hand, if the T cell secretes interferon gamma, then the B cell will become an IgG secreting plasma cell. Now, it turns out that every heavy chain locus, gamma, alpha, mu, epsilon, and delta, has 2 spare exons, one for membrane coding and one for secretion coding. So, for example, an IgM expressing B cell, which hasn't undergone isotype switching and isn't a plasma cell, has a constant mu region expressed with membrane-bound exon. Once the B cell CD40 gets bound by the T cell CD40L, an enzyme called activation-induced deaminase, or AID, gets activated and breaks the intervening DNA, removing all of the constant region between the VDJ and the decided-on antibody. So, as an example, an IgG secreting plasma cell would be left with a rearranged VDJ region, as well as the IgG region and the secretion coding exon. Alternative splicing will allow the cell to still have some surface-bound IgG, And that allows the cell to get antigen stimulation while it produces IgG. At this point, the cell is a fully differentiated plasma cell. Now, it turns out that most mature B cells will live for about 6 months in the lymphatic system and then die, without ever being stimulated by an antigen. A sad and lonely existence. To replace the dying cells, there's a plentiful supply of fresh new mature B cells coming out of the bone marrow. However, if the B cell does encounter an antigen and develops into a plasma cell, then it produces lots and lots and lots of antibody. Plasma cells live for about 6 weeks, and they don't divide. If you haven't cleared the antigen by the time the plasma cell dies, new B cells develop into plasma cells to take its place. Alright, as a quick recap. When B cells finish developing in the bone marrow, they use alternative splicing to co-express IgM and IgD on their surface. If a B cell finds its antigen, the antigen cross-links two B cell receptors, and that activates the B cell. If there's no T cell, it differentiates into an IgM-secreting plasma cell, also using the process of alternative splicing to allow for some of the IgM to be secreted and some to remain surface-bound. If there's a T cell nearby, the CD40-CD40L interaction combined with cytokines secreted by the T cell help the B cell class switch to produce IgG, IgE, IgA, or IgD.